I will be discussing how Frashella perrara increases the immune system activity in Apis mellifera and subsequently increases honeybees' resistance to infection. To understand the relationship between Frashella perrara and Apis mellifera, we must understand what a gut microbiome is. The gut microbiome is a collection of bacteria that have formed a symbiotic relationship with your body by aiding in nutrient digestion. Gut flora has also been shown to play a large role in immune system regulation in both humans and honeybees. Apis mellifera makes a great model organism because it is small, easy to breed, and has a very thoroughly researched life history and a fully sequenced genome. In regards to the immune system gut biome relationship, honeybees also display very important parallels to human beings while simplifying diet interactions and gut bacteria diversity. One particularly interesting honeybee gut bacteria is Frashella perrara, and it is known for its ability to elicit immune responses in honeybees. In 2015, Engel and his team further investigated F. perrara's effect on the honeybee gut. They noted as F. perrara colonies expanded into the pylorus, a scab formed. They theorized what they were seeing was related to a known honeybee immune response called a melanization cascade. This theory was supported by their findings due to an increase in frequency and intensity as bees aged. As in the figure, we see as a clutch increases in age, a higher percentage of the bees show the scab phenotype. We also see that the concentration of F. perrara in the gut concurrently increases. In 2017, Engel and a separate team sought to definitively prove that F. perrara was indeed the cause of the scab. In Figure A, we see a scab is accompanied by a present F. perrara culture in that same section of gut. They also demonstrate that no scab is formed when an alternate bacteria culture is present, as well as no culture present in both Figure B and C, respectively. And in that same study, Engel's team identified transcription and translation of genes relating to immune system response increased with the presence of symbiotic bacteria. All genes that experienced increased transcription with healthy gut biomes are shown by bars extending above the baseline. However, they noted additional increases in certain genes when F. perrara was present in higher quantities. These additional benefits are noted by the arrows under each gene that experienced the increase. In 2020, Airbon's research team started looking into European foul breed in Apis mellifera. They noted that individuals experiencing an infection with and without symptoms had higher concentrations of F. perrara and other gut symbiotes when compared to non-infected individuals. In the figures, we see that infected individuals, noted by the purple triangles and red diamonds, are sequestered together, showing their higher concentrations in relation to the non-infected individuals represented by the blue squares. The spike, they theorized, cannot be due to weakened immune systems due to its already established ability to increase immune activity, as well as its presence in healthy individuals. In summary, we see that F. perrara does indeed spark a melanization cascade that increases in intensity as honeybees age. This phenotype is related to increased immune system activity and gene transcription of known immune-related genes. And we see that the concentrations of F. perrara and other symbiotic bacteria also increase during active infection. The same team postulated that the increase is not due to secondary infection due to its natural occurrence in healthy bees and previously established benefits. With all of this considered, there is strong motivation to consider using F. perrara as a probiotic in an effort to bolster Apis mellifera's ability to prevent lethal infections. Now, if we were to increase use of probiotics in our cultivation of honeybees, this would have massive effects in both the agricultural realms as well as the human health realm. Agriculturally speaking, bees pollinate about 1/3 of all of our crops, and losing them as a main pollinator could lead to economic disaster, much like the Dust Bowl caused the Great Depression. Any research that helps increase hive stability is therefore of huge significance to human survival as a whole. In addition, anything that would decrease the use of antibiotics in both human and agricultural spheres is of huge importance due to the rise and threat of antibiotic-resistant infection in humans. Thank you very much for listening, and I appreciate your time.